So we'll continue from what we left with yesterday. So what was the thing which we were covering the other day? It was all to do with the solid and liquid side stream and how we get different types of side stream from both seafood as well as macroalgae, and we did not cover much of microalgae, but your macroalgae, shells, seafood, and what are the side stream. And liquid stream was mainly related to processed water and wastewater and how we can valorize that. So we'll and there was also a practical demonstration for shells valorization. So what was practically done yesterday was shells which colleagues from Rourkela, they got that shell, they washed it, they already dried it. What they did not do was the grinding part. Yeah. If you grind it, you reduce the particle size, dissolution becomes faster. And then this step was eliminated because if we employ ethanol, then you can get all those pigment. If you have seen the color, orange color or different type of color was there, and some pigments are colorless. So ethanol dissolves those pigments. So we did not do that, yeah, because don't wanted you guys to handle with ethanol because all are not from the practical background, yeah. And then what was done, you dissolved hydrochloric acid, and what other acid was used? Acetic acid, yeah. So if you, how many of you have added the egg directly to vinegar? If you add whole egg into vinegar, leave it for 24 hours, you will see the shell is gone, yeah. Because shell contains calcium carbonate, yeah. When it comes in contact with HCl, what happens? You would have seen some of the bubbles forming, yeah. That is basically a CO2 which is escaping. And what is formed in a liquid is calcium chloride, yeah. And if you have a food processing background, you know where the calcium chloride is used as a filtering aid, as a binder, another one. This happens with hydrochloric acid. If you use acetic acid, everything happens the same, and what you get is calcium acetate, yeah, right? And whatever is left over then is your protein plus chitin, because that is a robust network of minerals, proteins and chitin. So every shell does not contain a chitin. Chitin, protein along with the calcium makes very hard shell. That's why crab is having harder shells. Shrimp is harder, whereas other shells, what you see, mussels and snails, they have very soft shell because they do not contain any chitin there, yeah. So what happens after this? This step is called demineralization, yeah. So now whatever shells you will be seeing, it will be a bit softer. It may be possible it is not completely demineralized because of number of different factors. One of the factor is the ratio of it and the concentration of acid. That has to be optimized based on each and every species. So what ratio you used was 1:20, yeah. So if we use ratio of 1:50, it will dissolve much better. Can you guess why? Simple stuff is, if you have 1 kg of sugar, how long will it take to dissolve in 1 liter of water? Bit longer, yeah. But if you add it into 50 liters of water, it will dissolve straight away, yeah. So that is saturation level. That will be dependent on what is the saturation of end product into the liquid suspension, yeah. So that is estimated either based on the calculation or based on trial and error method. So these are the things which are optimized each and every step, yeah. So once you have this one done, you wash whatever residue is left over, yeah, to remove the acid. Then you add alkali, yeah. The moment you add alkali, all the protein will be dissolved in alkaline solution. Whatever is left over is your chitin, yeah. And this particular protein can be precipitated back with HCl to get a protein biomass coming out of shells. though it will be very small quantity. Majority of the composition of shell is calcium carbonate, yeah. And this chitin, if you mix it at very high temperature, it forms chitosan, and chitosan which is sold, yeah. So you have seen how the sequentially the processing is done, yeah. So maybe lecture 15 onwards, you'll see all the new technologies of usage of microwave and other processes, yeah. So that's the way it can be. Microwave, ultrasonics, high pressure, all those technology will help you to accelerate either this reaction or accelerate this particular reaction. Based on that, we deploy technology. Either we grind it. If you're grinding it, you are introducing energy into it. If you can use whole shell, it's much better, even though if it takes a bit longer. Grinding hard shell is very, very difficult, yeah. So we have done some of the innovative activities. When we have these shells, we sprinkle acid on it. As we dry it, the acid will weaken the shell. So the grinding energy is reduced, yeah, during the drying process. So these are all the interventions which can be done to improve this particular process. So this is your baseline process and how you improve your process based on technology, yeah. So if your monthly budget is 1000 rupees to spend, is up to you how you improve your quality of life. Either you spend everything in the first place, day one, or everything at the 30th of the month, yeah, or you have proper planning and distribute your funds so that your quality of life is different, yeah. So what we are going to cover today is what is duckweed, yeah. How many of you know about duckweed? It's not a weed which duck eats, but that's the name of the common name of a plant. Cultivation of duckweed and how we can use duckweed as a valorization tool for wastewater, yeah. So duckweed is actually a small aquatic plant, yeah, which is having a commercial cultivation mainly in US and Israel. We already discussed this. And you can get up to 30 to 40 tons per hectare and almost 55 tons per hectare, up to 55. So these are some of the statistics how you grow. And it can be grown on wastewater. What is microbiome? That is microbial microbes in biosphere. Diversity, stability with significant impact on duckweed growth and nutritional composition. So all these duckweed, whatever is grown, they have some unique features. And for that, any standard book or any standard aquatic plant book, you can look into it and you will find all those answers. One of the common species what I will be discussing further up is Lamina minor or Lamina gibba. So these are some of the species which are quite extensively used. So what are the best benefits of using duckweed? One, they grow rapidly. They do not compete with the crop or land because they do grow on water. They trap CO2 from air, lower energy consumption. That means you don't need any input fertilizer, easy to harvest and low cost biorefinery, yeah. So these are some of the rosy picture of duckweed. Now, if you'll see, how many of you have seen such plant growing in any natural water, yeah? If you look around your cities or any waterway or stagnant water, you'll see this type of plant pops up onto the surface. That is only because there is extra amount of nitrogen is available or fertilizer, leaching of the fertilizer from agriculture runoff or wastewater going into the water and it grows there. Either you will see greenish water, what you might have seen in, if you keep a water in a glass bottle and leave it out, you'll see some green algae is formed. That is basically a microalgae, yeah. And now how the microalgae is used now for range of different production. Same way, duckweed also grows. Think of the wheat crop which is grown for getting wheat flour or rice is grown. So how the wheat and rice was grown 500 years or 1000 years or 10,000 years or or a couple of thousand years ago. Yeah, they were all grown in a forest. People, they picked it. That is called selection. Those who have genetics or breeding program, they take those crops, they grow it in a mass, and then they do the mass production. Very similar approach is being now adopted for duckweed, microalgae, macroalgae, to see what are the beneficial compounds you can get, and then you start a mass production. Currently, two countries, as I mentioned, Israel and US, they are doing large-scale commercial production of these duckweeds. Yeah. So they all belong to family Laminaceae. There are 5 different groups, something like same as wheat, Triticum durum, Triticum dicoccum, Triticum monococcum. The different species are there, and there are not only 36, there are large number of species are there. Some they have a small bit of root, some they do not have a root. They are mainly a floating plant. They float on the surface. Yeah. So I'm not going to go into each of these details, but these are just basic details about duckweed. That means how much it doubles. Some doubles in 24 hours, some doubles in 3 days, some doubles in 4 hours. That means within 24 hours their biomass is doubled. They work on range of pH 5 to 9. That means you can grow them even in a wastewater of dairy plant because their pH is lower, or in alkaline condition, or on a neutral pH. So this is very similar what will be the condition for any other plant to grow. 6 to 33 degrees Celsius, that means that again depends upon what species we are growing. Yeah. So these are some of the factors which are again from different reference sources, and if you are interested in, I can provide you those paper, or I can provide you the comprehensive literature review on this one, which is already in a publication domain. So what I'll be focusing mainly on those things which you may not find it in journals or in articles on from real applications. So these are all temperatures required, sunlight is required, nutrient, and a pH. Yeah. So temperature naturally available, sunshine naturally available, nutrients from the wastewater, pH again depending upon the wastewater, or you alter the pH to grow that microalgae or to grow duckweed. So this is the way it looks like. Lamina minor, it's having small bit of root. Wolffia, Lamina gibba, Lamina porphyra, Lamina, there are a lot of different species. So as you can see, how we start growing in simple duckweed in a flask, yeah, how it is grown in a tank, yeah, and how it looks like, and how it is used. If you grow it in in normal water, it is still consumed in most of the places as a food entity. This species, Lamina or duckweed, they contains almost 90% water. They have only moisture in it, very less amount of solid. Since they have a vacuole, so that's why they float on surface of water. So how you can produce it? Even if you go in natural water, pick that plant, put it in the water, and you will see slowly it starts growing. Yeah. It's something like you bring in any plant and start planting it, and then from there you grow multiple plants. There's no rocket science in cultivation of duckweed. Yeah. So as you know, all the scientists, how we are all known to complicate things, then you start looking into growing in different types of water, grow it in a milk, grow in a spoiled milk, grow in clean water, then you'll see which one is better. Yeah. Based on that, you develop your own process, you get something interesting, you write a report, you publish it, that's the way it goes. Yeah. Or else you look into the literature, what people have done, you repeat it, it's just the copycat. Yeah. So that's the way how innovation works. So that's all simple duckweed, how it starts in lab production. Now these are some of the complicated or advanced way of growing duckweed. As you can see, this is the top portion of duckweed, normal tank. You have dissolved oxygen, oxygen reduction potential, all these measurements are carried out. And this particular study found if you do have the aeration in the tank, even though you aerate it when you do wastewater treatment, Yeah, aeration is required. So when you aerate it, their growth is much faster and their nutrient recovery from the wastewater is much higher compared to non-aerated one. So the intention here and our objective in this whole of the lecture module is how we can use this duckweed to remove nutrients A or to reduce BOD and COD of water and use that particular wastewater to grow a new type of biomass. Yeah. So as you can see from here, these are some of the tanks which are cultivated, and this is the open cultivation or a closed cultivation. That all depends upon which country you are growing in, is a high temperature or a low temperature, because the temperature requirement is different. So it is very similar to what way you have a wastewater tanks. And then in our Israeli industrial partner who is growing duckweed, they have changed the color of this duckweed from green to red by using a specific nutrient which is absorbed by a plant and becomes red. The purpose of having that red duckweed is to use as an alternative meat so that you get the same color, protein and everything. Yeah. How it can be made red? If you buy a white rose or a white lily, if you put that lily into red ink, what happens? All the white petals become reddish color or bluish color depending upon the ink. That means dye is taken up. Yeah. And if that is having a betalain or if having a beetroot juice or any of those things, that pigment or anthocyanin pigment is taken up by these plants. Yeah. So you can change the color. Then you dry it, you have same red color. That was a very, very simple innovation, but it's very interesting. Yeah. So it was not that somebody discovered a rocket or somebody discovered a new equation how to launch a rocket. Yeah. So that's the way you can see from the image how we can grow duckweed. So these are again a lot of new ways of doing studies and production are ongoing. So as other day I was mentioning about how we are cultivating at a larger scale. These are all for demonstration site, as this is Mount Lucas in the center of the country, and here as you can see these are flowing water. This is not a small land, this is a couple of hectares of land. These are flowing water and they have a fish in different places. Yeah, fish is moving in this water and duckweed is grown on the top. All this is exactly what you see over here. Yeah, this duckweed is growing and also the energy is taken from wind, that's wind turbine. So everything, this is as a demonstration site to improve circularity. This is one of the place called peatland or also known as bogland. So that is basically the soil is having so much of carbon, you take a soil and you can use it also as a fuel because there's a lot of embedded carbon. And as you can see, this is all duckweed and this is the man who is just collecting duckweed from the surface. Yeah. So that means fish is also consuming duckweed as their feed and fish is also produced within that. That's trout. Yeah. So you get both the things, how you can make a complete circle. So as you can see, now this is what open land looks like. This is just a normal trawler which is just aerating it, and that can also be used for collection. As you can see, there are a lot of other type of weeds are also grown. These are also aquatic plant. Then some of the plant operators, the people who are working there, then interest came in about this species. Let's see what is this species. Yeah. So that's the way how things move. So there is also issues with this particular protein. When you are growing in a water, there is also a possibility of shells growing in it. So you may have shellfish allergens in it. So it again, you have to be careful of all those nutritional labeling or allergen notification, whatever allergens it might have. And this is completely controlled cultivation. Yeah. And same cultivation can be in a moving water, how you treat it. Yeah. So this is open pond system and this is closed. So how difficult is to grow duckweed? Go to the river or pond, pick one plant, put in the water, in 2-3 days it will double. Then from there you start cultivating. From one seed, one wheat seed, how many seeds you can get? After 3-4 months, you can get more than 1,000, right? And from 1,000, you know how much you can get, right? The same thing works here. So there is no seed for this one, but whereas a small inoculum or can be done with the spores and different way, like what when you see the seaweed cultivation, macroalgae cultivation, you will be able to find that. Where all we can use it and how we can grow this. Clean water is easy, yeah, but that can also grow in slurry, animal dung slurry, food processing wastewater, swine, because you know wherever swines, that's pig farming is, you have all those lagoon effluents are there, you can grow all those duckweeds there, yeah, or any of the wastewater. They all have a problem of high BOD and COD. You adjust a pH depending upon species you grow. Nutrient recovery, you may, if you are looking for a large amount of production, then you may have to see how you can adjust calcium, magnesium and all those ratio, and that can again complement from the experiment what you were doing the other day, yeah. In some cases in agriculture, eggs, normal eggs or rotten eggs are buried into the soil. For what? So that calcium and magnesium, all the nutrients are embedded into the soil and crops can take those, yeah, rather than supplying external chemical fertilizer into the soil, yeah. And then this is the way duckweed cultivation can happen, and then you get a biomass which is rich in protein. It removes the nutrients from the wastewater. That means inorganic form of nitrogen, that is nitrates, nitrites into proteins, yeah. Carbon forms from CO2 from the air to make into starch, cellulose and the nutrients. So that's what it is, yeah. Is it complicated to grow duckweed? It's very straightforward, right? So if you'll see how much duckweed can remove or can be produced, slaughterhouse wastewater, that is animal abattoir, 330 grams per square meter after 30 days. This is dry mass. Oil palm effluent, just depending upon what they are reported in different studies. Chemical oxygen demand for the wastewater reduced by 18%, CO2 85% after 28 days. It's just again depending upon what is the intention and how different types of water can reduce. So this is the way you can use your own wastewater and cultivate it. And remember, this is all the agriculture wastewater. Think if you are involved in protein extraction, you have a lot of wastewater or processed water generated after protein extraction. Like what you have done the experiment, you have acid solution. If you remove the calcium chloride either by membrane or even now you will be going to do drying part, that water, you are going to dump it. That water is having some of the nutrients and that have a specific pH. You can select a strain to grow duckweed on it, and that's the way circle can be completed. So that works on circular economy principle. That means closing the circles, yeah. So that's one. Once production is there, harvesting. Simple scoop. The way you take a cream from the milk surfaces, yeah. You do mechanical harvesting or you harvest it at a regular interval depending upon how much biomass is produced. So it's a continuous process. That means wastewater keeps coming in, yeah. You keep recovering your duckweed. So it's not that the wastewater is there, only certain amount of nutrients are removed, then you dump it back. So it can be a continuous process, continuous supply of wastewater, continuous production of biomass and continuous harvesting. Did you get the purpose right here? Yeah. So if you have a large area, so you are continuously getting biomass. Whereas if you are using the wastewater treatment system, what you are getting continuously is the sludge, which has to be disposed at some point, some way or the other. Then again, wherever you are disposing, then you have aluminum and all those chemicals which are going in. So with this, you are not adding any nutrient or you are not adding any input chemicals, because when you add any chemical, flocculating agent or so, there is also a cost and environmental impact of producing those chemical is also associated with life cycle assessment. Yeah. So 6 to 7 grams dry mass per square meter per day. Yeah. So it's a dry biomass, because wet one will be quite high. And what type of protein this contains normally? The protein in this one is mainly Rubisco. So normal protein content is around 25 to 35% in this duckweed on dry basis, and half of that protein is Rubisco. So you don't need to use any alkaline extraction or anything. Simple squeezing of its juice, precipitating the protein, you get more than 50% protein in it. So you can classify it as protein concentrates coming out of it. It also contains starch, it also contains fiber, it also contains different bioactives, and those bioactives, they do have antimicrobial properties, and there are some IPs which I cannot go and discuss over here. Some of the bioactives from this particular plant are also used as a biofertilizer for different agriculture crops. Yeah. Anything which grows in very high amount of water, they have very good antibacterial property, because they do grow in a water, and bacteria needs higher amount of water to grow. So that is whole open area of research to explore, yeah, and do something interesting, something new. Otherwise, doing the same bit of things what are already there in literature, already people are doing, something like keep chewing the chewing gum, even the sweetness is gone. Either you can't swallow it or can't spit it, yeah. So duckweed application, this is again is taken from, this has been taken from CGB bioenergy, duckweed, fermentation, the bioethanol, biogas or biohydrogen production, because it contains starch. Once you get a protein, left over is fiber and starch, that's again for ethanol production, can be easily used for fermentation. It contains starch, so there can be a number of different applications. All these applications are non-food application, just because when we are using a wastewater, you have to go through the regulation for food application. Yeah. So you have a lot of non-food applications. Then you have flavonoids. A different type of compounds can be formed. That is again bioactive compounds. You have protein for both animal feed as a protein powder. And these are some of the brands which are sold as the water lentil. What is the lentil? Masoor. Yeah. What's the botanical name? Lens esculentus. Yeah. So same way, this is water lentil powder. Then again, other chemicals for different applications. Yeah. So when we see this, automatically we think, oh wow, this is very nice, I should work on this. Yeah. Then you go to the next presentation, you say, oh, microalgae is nice, let's do this one, right? Remember one thing, other side of the river is always green. Yeah. So whatever you do, just be concentrated on that one, and there will be always the hurdles comes in between. Clearing those hurdles is the best part of it. Yeah. Never to jump on a topic, because you are here listening to duckweed. Tomorrow it's water hyacinth, you'll move to water hyacinth, then you move on to seaweed, then you move on to this one, right? There's a saying in Hindi, what you call, dhobi ka kutta ghar ka na ghat kar. That means neither you can complete any of the work properly. So just whatever you're doing, just learn the approach. Yeah. Learning the approach is important. Yeah. Dressing like somebody is not copying. Buying exactly the same dress is a copy. Yeah. So remember, that's not to get so intrigued about it. But if your focus is only on production of bioethanol, and that time you can consider different crops. Yeah. If you focus in a valorization of wastewater, then you can consider different crops. It depends upon where you want to use all this knowledge. Now, Coming back to composition, so as you can see, this is 36% protein, this is gram per kg. Always be very, very careful with the units. Units are mix and match here. Say milligram per gram of protein, yeah? And only 3% fat. So it's again having saturated, monounsaturated and polyunsaturated fatty acids, some of those minerals. Now, when you see the lead, aluminium, chromium, all these things are coming in from the water. So that's why these plants are also known as phytoremediation. That means you capture those heavy metals from the water to clear the water, yeah? And then that's second question, what you do with this biomass, yeah? If you're concentrating it, getting only the ashing, so you're concentrating the minerals, yeah? It's not like in nutrition they say if you have iron deficiency, that does not mean you just go and start eating the iron rod, right? There is a biological form and there is chemical form. Hydrochloric acid is also produced in human being, in human stomach, but that does not mean you start consuming the hydrochloric acid. There is a difference between biological origin as well as mineral acids, A. B, when you see arsenic, how many of you know arsenic is dangerous? We know arsenic is dangerous, yeah, heavy metal. But the problem is naturally occurring arsenic or chemical form of arsenic, yeah. We do not have a method to segregate that. So that is natural occurring one is still okay compared to chemically processed. So these are some of the differences what we have to understand. And as you can see, everything is compared against soya, because in Europe we are looking only alternative for soya and they have a quite similar or a better amino acid profile compared to soybean. And this particular data is again depending upon the species or mixture of species. This one is what was calculated or is estimated from our lab from that particular site, but that particular site is not having a single species. The Labna minor, Labna gibba, the multiple species are there as a whole biomass. Those who are working in alternative protein, you would have come across on different research about leaf protein, grass protein, yeah. Our research group is also working on a grass protein, which is all in a garden grass, because we have plenty of grass. The massive amount of green grass is there around the year because it rains most of the time. It's having a grass, and to extract grass protein, a very similar principle applied for duckweed to extract a protein, because majority of the protein is rubisco and several enzymes, yeah. And rubisco is very high molecular weight protein. So what should be the procedure for extracting proteins? How do we extract protein? Anybody?
So alkaline extraction and isoelectric precipitation.
Okay. So what makes us to use alkaline followed by isoelectric point or isoelectric precipitation? Why alkali? Why not acid or water?
Sir, alkaline helps to maintain the particular pH.
Okay. Well, that's true, but one thing is, before we select a solvent, this I will be repeating again when we go into the technology. One thing what you have to do is, first you have to do the protein profiling, yeah. It is very similar. If you want to take this whole table out of this door, what you have to do? First measure the size of the door and measure the size of the table, then you have to take horizontal or a vertical, right? Very similar way, before starting extraction of protein, alkaline, it's just because majority of the cases proteins are soluble in alkali. You have to do one thing called Osborne classification. How many of you have heard this word? Osborne classification. I think everybody whosoever has studied basic biochemistry would have studied this. Yeah? That is solubility of protein. Yeah? Protein is either soluble in water. Yeah? So those who are making egg alternative protein, fish alternative protein, meat alternative protein, they have to know first about this Osborne classification. Otherwise, it will be just wild guesses, cook and look type research activity. Osborne means one, certain proteins are water soluble. For example, albumin, globulins are water soluble. Yeah. Some proteins are salt soluble. That is 0.5% or 0.2 molar or 0.5 molar, something like that. 0.2 to 0.2 to 1% salt soluble proteins are there. Yeah. And then if you increase the salt content, protein precipitates. That's called salting in, salting out. You take some amount of salt to dissolve a protein, increase the salt concentration, protein will precipitate, right? 2. Third, alcohol soluble protein. Have you heard of it? Yeah. 4th, alkali soluble protein. 5th, acid soluble protein. Now in fish, acid soluble protein is your gelatin or collagen. Yeah. Majority of the protein is alkali soluble. Some is alcohol soluble, some is salt soluble, and some is water soluble. Just now I mentioned about duckweed protein, majority is Robisco. It's a water soluble protein. So if you use an alkali, your yield won't be that great what you get it from the water. Yeah. So without that, using just a common solvent will not solve your problem. Yeah. Okay. So that's why if you do Osborne classification, then you know what type of protein is there in a biomass. Luckily, you don't have to do it because majority of work is already there in the literature. But I always advise any of the guys who within our group working on a protein extraction, they do this first. Because this is the important exercise to get different types of protein. For example, you would have seen some of the patent as egg replacer proteins from mung bean, yeah, by using cross-linking with sulfur or some cross-linking along with it. Because mung bean protein is a water-soluble protein and it's just exactly, it solubilizes easily, yeah. So that's the way you can get all the different type of water-soluble protein, fat-soluble, water-soluble protein, acid-soluble, alkali-soluble, depending upon what is your application. If you are making a beverage, if you want to add protein to orange juice, so you can only add that much amount of protein which is soluble at that particular pH. Yeah. So basic background of any of those solution or solvent is very important before you start thinking, let me extract it by using ultrasound, microwave or anything. Yeah. So majority of the protein is alkali soluble. That's why we think alkali is the best solvent. Yeah. Now, this is lamina minor, fresh duckweed. When you freeze dry it, you get a perfect green powder. Yeah. If you sun dry it or you hot air dry it, still you get slightly darker color. Yeah. This is what normal most of the experiments are carried out. Then you have different types of technology by using normal stirring. What you do most of the time, if you add alkali or water to extract it, or use ultrasonic systems or use high pressure. I just giving a brief example how the protein is extracted from duckweed first, then starch or a fiber. Or else if you take, this is the normal protocol what you would have followed in your experiments if you are doing the experiment. What I would do differently, I'll take this, I'll get the juice out of it. Use a very nice juicer to squeeze all the water. Yeah. When you squeeze all the water, what you will get? All the water soluble fraction, and you leave it for some time, you will see starch precipitates at the bottom. Yeah. How many of you know how to extract the starch? Again, very simple. If you peel a potato, you will see white color powder. What is that? That's the starch. Yeah. That means starch is embedded like a globules within a matrix. Yeah. Because that's a storage. It stores all the glucose in the form of starch. The moment we get a juice out of it, you leave the juice for a certain time, all the starch will precipitate. Liquid portion, if you dry it, use a membrane, you get your protein. The starch and protein is gone, then whatever is left over protein and a fiber is now your alkali soluble protein, then you can use alkali extraction process to get the second round of protein. Yeah. That's the way a biorefinery processes are developed. Yeah. Remember the sequence. What should come first depending upon what is larger in quantity, or remove what might cause a problem with further extraction. That means you have to remove the lipid before you extract alkali protein. The reason is if alkali adds with the lipid, it forms soap. Then you will make soap. Yeah. So we'll come back to these technologies again. Yeah. These were all fancies of one of the PhD wanted to use all those technologies. Same thing like you all guys look into the paper. Oh wow, high pressure. Let me try ultrasound. Let me try this, and this is what you end up with. Yeah. But there could be a simple methods. You don't need a computer to get 10 plus 10. You don't need a calculator for that. Yeah. So this is one of the example here. Now, yesterday and day before yesterday, I was mentioning about Bioeconomy Student Challenge Award, which each and every country was sending one of those ones. So we had a team of our one of the PhD student, 2 masters and 1 undergrad. They all went for this one. They kept its name as a phyto towel. Yeah. Whole logic was dairy is a big industry in Ireland. Yeah. And huge amount of dairy is exported. Think in a country there was a milk quota. That means you cannot produce milk more than this quantity because it was very difficult to use it or export it or to diffuse it. Yeah. So large amount of milk goes for a cheese, and then they develop this type of phyto stacked system. So far whatever you have seen is single layer or a big tank, but you can do the same thing in a stacked tank basis. Mobile, modular, plug and play, scalable and fully automatic harvesting system. That means if water is moving, yeah, so you can have some strainer. The smaller duckweed can pass through and larger will stay there. So that's the way you can continuously harvest it and continuously reduce the BOD and COD in this particular chamber. I'll quickly go through this so that it will give you a clear idea what I intend to say here. So this is your all the light system, pipe, filtering conveyor, water catchment, biomass holding tank and pipe in, pipe out. And if you want to make it further more automated, you put a sensor here to measure its BOD and COD, and then again your machine learning, that means you fit in with an equation, yeah, to say that when BOD is less than 50, it should be discharged. Otherwise, it continues within the same circle. Yeah. So, simple idea from beaker, how you can grow duckweed. You can make as complicated design as you want. Same thing, normal phone was 1G, then 2G, 3G, 4G. Yeah. What's the difference between 2G phone and 5G phone? And that is very important because this topic will come in again. You know any difference between 1G, 2G, 3G, 4G, 5G or GIAN? Yeah? Do you know the difference? We all say, oh, I have 5G network. Do you know what a 5G means? Yeah? 1G was basically when you can only make a call. Yeah? First generation. 2G, phone and messaging. 3G, phone, message and internet. Yeah? That means all three can work together. In 2G, you can also get an internet, but when you are on a call, internet disappears. Yeah? right? You have noticed that one. 4G, all three will work together at a higher speed, and this was very, very high speed. Yeah. So that was the only generation difference. Because obviously, when you are on a phone, you don't need an internet. So that's why all these generations were there. And you'll see this reference of this one when we go through the protein extraction process. Yeah. So this is what I have referred also as a protein, first generation protein, eating normal meat, pulses, legumes, that were first generation. Second generation, whey protein, all these, whey protein isolates, protein concentrates, second generation. Third generation, your insect proteins and all new sources of protein coming in. Fourth generation, how we are going in changing our protein in our diet. Earlier, what was it? You eat a protein-rich food, healthy. Now you need a protein supplement. Generations, first, second, third, fourth, fifth generation. That's why we classify as protein juggernaut. Yeah. Now coming back to this, so this was a simple design. This is the repeat of the slide. That means either you go for conventional or go for simple circular system. Yeah. So this image what you saw in 6th or 7th lecture, this was the history as now we are going through. Yeah. So sometimes I do take movies approach like a Bahubali, first and then the conclusion comes in, right? The same thing. So something happens and then you go back to the history so that you won't forget it. Yeah. So how this all came in, all the way from simple beaker, and for that you don't need to have a biology degree to understand how to grow a crop. Yeah. Farmers, when they grow a crop, they do not study agriculture or agronomy. Yeah. They learn by a practice and then to find the answers to the questions. In search of that, you develop scientific principles. Yeah. Because this could be a controversial statement. Science is always reactive, yeah, not active. Things were happening, we draw theories based on that, and then we say, based on this theory, this happens. Yeah. So what was happening? Apple was falling before the gravity was discovered. Yeah. Correct? It's not that when gravity was discovered, then only apple started falling. So sometimes there is always an, oh, you believe in this and you are a scientist. Yeah. Right? So science is always reactive. My name is Brijesh because my parents calls me Brijesh. If they would have called me tomato, I would have been known as Tomato Tiwari, right? So that's the way the processes and everything is developed. So anything, whatever happens, you find an answer to it, you develop an equation, and then you believe it is always going to be this. If you would have studied the first order kinetics, yeah, every growth is exponential growth, yeah, microbial growth or anything. It cannot happen if microorganism is growing, all of a sudden it dies and then again grows, yeah. So that means everything will go in first order. First order because it will never touch zero. How many of you have studied half-life? What is a half-life?
So reduce the radioactive by half, 90%.
Okay, by 50%, yeah?
Sorry, yes, sir, sorry, sir, 50%.
So that means 100% will be double of that. Why?
Like in next cycle, it will again reduce 50% of it.
It will never go zero, right?
Yeah.
Why? Because it's first order.
Yeah.
It's very simple. It's nothing. I know your formula is 2.303 upon k. Yeah, but think exponential equation is what? Population at any time t, yeah, is equal to initial population, yeah, exp minus kt, yeah. Same thing, concentration at any time t is equal to or directly proportional to initial concentration exponential minus kT. Why minus? Because it is reducing, right? So you said half-life, that is Nt is equal to 1 by 2 or N0 by 2, right? If you put N0 by 2, N0 exp minus kT, yeah, gone, 1 by 2, now this 1 upon exp kT, that's why we use minus, yeah. In math, signs are used, so remember data is for maths, math is not for the data, yeah. Thing happens, we fit an equation. If you fit a best equation, that's the way model works, yeah. So that means exp kT is equal to 2, is natural log, yeah. ln of k, then if you take natural log both the side, this becomes kT, yeah, is equal to ln 2, correct? T, half-life, ln 2 by k. ln 2 is what? Natural log of 2 is 0.693. Simple, yeah? And this is what we did study when we were 8th or 9th class. Half-life is this, yeah? And we always remember this formula. Amount of time required to reduce the population by 50%, yeah? Next 50%, that means it will be like this. That means it will never go zero. That means nothing can be destroyed from this earth. Back to the thermodynamics law, that means we use exponential one. And moreover, log of this is not defined. That's the only reason why we use these type of numbers. And same thing happens with the, why I'm touching this one again, because when I go through the technology section, my life will become bit easier, yeah. And you may have some more questions coming in. So how many of you have studied microbiology? What is the D value? In a mic here, please, yeah.
Sir, the time taken to reduce 90% of microorganism at a definite temperature or constant temperature?
90% population, yeah. That means 1 log cycle.
Yes, sir.
So 2 log cycle will be?
Sir, 2 log cycle again, next one, sir.
Tell me how much?
Sir, next 90, I guess.
Yeah, that means if I have 100 microorganisms, 2 log will be how much? 90 or 91 or 99?
98.
It will be 1.
So 100 becomes 1, yeah. 2 log means 99% removal, right? 3 log 99.9, 4 log 99.99, yeah. In the same way, food processing comes 12 log, yeah. That means 99 point so many nines, but still not 100%, yeah. Now, why we use log number in microorganisms? We say no, log 5 log, yeah. We have only 2 log reduction. 2 log reduction means 99% of population is removed. But still, if you take any of the food processing book, you need to have a 5 log reduction, 6 log reduction. Why? Pasteurization is 5 log, sterilization is 12 log, yeah. Why do we use log number? Why don't we just write it directly how much population is there? Any idea? Again, simple assignment. Think over it when we go into the technology for reduction. That time we'll go to that one. Yeah. Why do we use log number for microbial population? Yeah. Why not actual number? Population is 3, 7, 8, 9, 4, 6 bacteria. Yeah. And we use unit called CFU. That is colony forming unit. In one bacteria can form one colony, one bacteria can form 1,000 colonies. Yeah. Other way around. One colony can contain one bacteria and the same colony can contain 1000 bacteria. Something like Delhi is also a city, Rourkela is also a city, yeah? Population is different, but city is something like CFU, yeah? Come back with the argument, why do we use log number? I'll make it further more complicated. One of the argument could be it's a large number, that's why we use log numbers, right? But have you ever seen your blood reports? 5.9 into 10 to power 6 cells per ml. Why we do not use log number there? WBC, RBC, all these blood count and all other counts of human body, they are exact numbers but not log numbers. Think over it, yeah? And if you find it in any of the journal, let me know which journal has written that information. That is all is there in basic books which we often ignore. So coming back to this one, this was all the pitch, high protein for animal feed in duckweed, environmentally destructed animal feed, if you are using soybean crops, because it is not really environmentally destructive, but at this stage it is, because we need more and more land to grow crops. Population is increasing, we don't have a land, so start cutting the forest. So we are disturbing environment. Whereas for this, you don't need land, yeah? So whatever all this fertilizer, when you cultivate it, you have again environmental degradation, whereas all these type of phosphorus, calcium and everything is recovered from the wastewater and goes into duckweed which contains those minerals. So that will supplement. Yeah. Now, first question, duckweed is easy to cultivate, yeah. Second, processing is very simple, you just dry it and use it directly for the feed purpose. Or duckweed is grown, if you have a fish farm, same duckweed is consumed by the fish directly from there. So you don't have to go and make a protein for fish feed. So this is called circularity. That means to introduce the short circle, yeah. You cannot achieve complete zero waste, but that's the way we can get it. Now I'll quickly go through again with the practical. Some of you were not there present in the beginning, yeah. So basically what experiment was done yesterday was this part, yeah. You had the shells, yeah. Who's going to repeat it for me? I am already bored now. Who was awake when I was doing this? Who want to repeat it what we did yesterday? Or you did yesterday? Okay, I'll repeat it anyway. So shells were washed and dried, what you got it, yeah? And we eliminated this particular step because don't wanted you guys to handle ethanol, and this would have taken away your astaxanthin and some of those pigments which were colored, yeah. Alcohol can remove most of the pigments out of the biomass. If you use a methanol, even better, it can remove those pigments. Ethanol, methanol, diethyl ether and all those solvents, whatever you guys were telling me, all those solvents can be used to remove those pigments. Then we are adding organic chemicals, and subsequent food and non-food application becomes trickier. So we eliminated this. So you had your hydrochloric acid, mixed it, and duration can be overnight, 2 to 3 hours, depending upon the concentration, solid liquid ratio, temperature usage, stirring, non-stirring, all those things will dictate how much time will be, that has to be optimized. And when we use hydrochloric acid, what is the composition of shell? Calcium carbonate, chitin plus protein plus others, yeah. The moment we add HCl, calcium, and this is broken down into CO2, goes into the air, and you get CaCl2. You can do the mass balance very, very easily. You know how much calcium is there in a form of calcium carbonate? Get all your chemistry books opened, simple analytical chemistry or physical chemistry. Calcium chloride is formed in liquid suspension. Leftover biomass is chitin and protein. That means you are at this stage, and today your instructors will show you to wash it, and then they will add alkali, and this alkali will solubilize the protein. So, protein is out in one phase, calcium is out in one phase, and then if you leftover biomass will be chitin, and this chitin can be converted into chitosan, or even if this chitin is added to the soil, it will act as a biofertilizer because this chitin contains nitrogen. Even after demineralizing it, whatever is left over here, protein plus chitin, is also a sort of biofertilizer because it protein contains nitrogen that also acts as a fertilizer. Yeah? Make sense? Or half demineralized frustration. That means you do not have to complete demineralize it half, so that you have a calcium, magnesium, everything added to the soil. Then it becomes easier to handle in agriculture field, or whole shells can be added directly to the crop. So, those are some of the ways these type of biomasses can be used.